Welcome to the Torrey Botanical Society Torrey Talks lecture series. I'm Catherine Mercier. I'm the programming chair. And I just want to give you a little background about the society before we start. So the Torrey Botanical Society was founded in 1867 um, by collaborators and folks related in some way to John Torrey. And the mission of the society is to promote and disseminate information on all phases of plant science. And to that end, we hold public lectures like these. We also publish the Journal of the Torrey Botanical Society. I think the second issue for the year may have just come out or will be coming out soon, so check that out. We also have free walks. So we have a bunch of upcoming walks actually. Make sure you check the website for those. This Sunday, we're going to Cunningham Park in Queens. The Sunday after, we'll be in Manhattan at Fort Washington Park. The Saturday after that, we're in Staten Island at Clove Lakes. And then the following weekend, the 23rd, we're in Rye. I think that's outside of the city. But make sure you check out the website. There's a bunch of upcoming walks. Springtime is a great time to go outside and see some plants. We also have undergrad and graduate student grants, as well as grants to hold like host symposium at conferences. Um, and if you want to know more about the society, feel free to become a member. It really is, you can learn about everything that's going on, and it's really helpful to the society. Get all the journal articles, get all the emails about the stuff that's going on. Um, so we You can also follow us on social media. We're at Torrey Botanical on Instagram, where we're becoming a little bit more active on Blue Sky. We also have a YouTube page where we post recordings of all the lectures. We're at Torrey Botanical Society on YouTube. And if you want to share this talk with somebody after it's over, feel free to, we'll get it up on YouTube and you can share it that way. A couple notes about the Zoom before we jump into the talk. Please stay muted during the talk, but you can feel free to have your camera on or off. You're willing to put questions or comments into the chat, and then we'll have, after the talk, time for some Q&A to address the questions that were put in the chat, all that good stuff. And with that, I would like to introduce Tanner, who is a graduate student at the University of Georgia working on mountain mint. Tanner reached out to the society because he is working with one of the specimens collected by John Torrey. And so I think that's a very nice connection to the society. Um, and today officially was was a master's student whose work has expanded so much. Today is officially a PhD student. Uh, blowing up your news, Tanner, there. But exciting stuff. And with that, I'll hand it over to Tanner to talk about Mountain Mint. Thank you so much for being here.
Of course. Thank you so much for having me. I appreciate the introduction. As y'all know, my name is Tanner, and I'm a grad student at University of Georgia in the Rooter Lab. Now, today I want to start off with what seems like a pretty simple question. What is a mountain mint? But as I've gotten deeper into this group, I realized that it's not actually a simple question at all. Mountain mints, or Pycnanthemum, are a group of native plants found primarily in eastern North America. They're well known for supporting pollinators. If you've ever seen one in bloom, it's usually covered in bees, wasps, butterflies, all kinds of insects. They're incredibly active plants ecologically. But from a scientific perspective, they're also kind of messy. It's not often clear where one species ends and another begins. You can have plants that look very different but are classified as the same species, and others that look nearly identical but are considered separate. And that raises a lot of questions about how we define a species within this group and what's actually going on genetically and even evolutionary. So in this talk, I'm going to walk you through how mountain mints vary across the landscapes, what we're seeing in field collections and garden studies, and how we can start to make sense of these blurred boundaries. And ultimately, how can we take something that's already ecologically valuable and begin to understand it, and maybe even improve it? So, before I get too far into the science, I want to take a step back and explain how I got here. My interest in native plants really comes from a combination of ecology and horticulture. I've always been drawn to plants that aren't just visually interesting, but that actually do something. Plants that interact with their environment, that support insects, and that fit into a much larger system. And mountain mints are kind of the perfect example of that. On the left here is a close-up of one of the flower heads of Pycnanthemum, and you can just see how intricate these structures really are. On the right is what looks like a whole new level plant in a garden setting. They're not just ecologically functional, they're actually really strong ornamental plants too. They're tough, they're adaptable, they perform well under low input conditions. And trust me, we get a whole lot of that here in the Piedmont of Georgia. And they bring a huge diversity of pollinators. But despite all of that, they're still relatively underutilized in horticulture. And part of that comes back to the same issues I mentioned earlier. Uncertainty around the species identity, variability, and how consistent these plants really are. So for me, this work really sits at the intersection of two things. Understanding these plants from a biological and evolutionary perspective, and thinking about how we can use that knowledge to make them more accessible and widely used in managed landscapes. And this is where that starts to become really relevant on a broader scale. There's been a huge push in recent years towards designing landscapes that support pollinators, and for a good reason. We know that pollinator populations are declining and that plant choice plays a major role in supporting them. So you see more and more plantings like this, diverse, layered, multi-species systems that are designed not just for aesthetics, but for ecological function. Now, native plants are a big part of that conversation, but not all native plants are created equal in terms of how they perform, how consistent they are, and how easy they are to work with and design landscapes. And that's where mountain mints start to become really interesting again. They're consistently ranked among the top for pollinator visitation, and they're incredibly attractive to a wide range of insects. But at the same time, there's still a lot we don't fully understand. So if we want to really integrate plants like this into design systems, whether that's better understanding of, whether that's in public gardens, restoration work, or even the nursery trade, we need a better understanding of what we're actually working with. And that's where my lab and my work really starts to move directly into plant breeding. So what you're looking at here is one of our field plots at the UGA Horticulture Durham Farm. Each of these sections represents different accessions, species, or populations of mountain mint that we're evaluating side by side. And this setup is really important because when you're working with native plants, you're not just starting with something that's already been highly selected or standardized. Instead, you're dealing with a lot of variation. Different plant sizes, flowering times, level of vigor, and even differences in how they respond to stress. So breeding in this context isn't just about creating something new, it's about understanding what variation already exists and figuring out which traits are actually useful or desirable in a landscape setting. And that's especially challenging in a genus like Pycnanthemum, where species boundaries are already unclear. Because before you can even start beginning to improve something, you have to ask, what exactly am I working with? Are these consistent populations? Are they mixing? Are they stable over time? So a lot of this work starts with observation and evaluation, really getting a handle on how these plants perform across a variety of environments over and over. And over multiple seasons at that. And that starts to become the foundation of everything that happens next. So as much as I love native plants, there's a challenge when we try to use them in design landscapes. Many of these species just haven't been selected for ornamental performance. So what we often see instead are traits like lodging, where plants flop over and lose structure, defoliation, whether from stress, insects, or seasonality, and just overall poor habit, where the plant just doesn't have that clean, intentional form that people expect in a garden setting. So while these plants are incredibly valuable ecologically, they don't always meet the expectation of horticulture or the nursery industry. And that tension between ecological value and ornamental performance is really where my work begins. So the point of this slide is to really outline the trajectory of the story I'm going to tell today. So what we have is a pretty simple question. What is a mountain mint? Now, at first, that seems pretty straightforward. You can grow a plant, you can observe its traits, and you can even try to assign it to a species. But pretty quickly, things start to get more complicated. We start to see variation that doesn't quite fit the descriptions, and you start to see plants that look intermediate or populations that don't behave consistently. And that leads to the second part of the story. Really, what is going on with this genus? Why is there so much variability? Are these species actually distinct? Is there hybridization happening? Are we looking at stable forms or something a lot more dynamic? And then finally, that leads in what do we think is the most interesting piece. It's not just a taxonomic puzzle, it's also a conservation problem and a breeding opportunity. Because if we don't fully understand how these plants are structured genetically and ecologically, it becomes harder to conserve them effectively. But at the same time, that variability might actually be something we can use. It might give us the raw material to select for better ornamental traits, improve performance, or even new forms that haven't been quite developed yet. So the rest of this talk is really about connecting those pieces, taking the system that's a little bit messy, and trying to make a sense of it along the way that's useful for both science and practicality. So with that, the first part of our talk, what is a mountain mint? Now, for those of you not so familiar, mountain mints don't just exist in isolation. They're part of a much larger family, the mint family, or Lamiaceae. And this is a really familiar group of plants like lavender, sage, thyme. These are just some of the plants that people recognize immediately, whether from gardening, cooking, or just general use. And what's interesting is that within the family, we see a huge range of diversity. You have plants that are highly cultivated and very uniform, like lavender, cultivars that have already been selected for consistent flower color, compact growth, and uniformity. And on the other end, you have a genera like Pycnanthemum, where there's so many mountain mints that are kind of just sitting in a space within a very well-known family, but they haven't really gone through the same level of domestication or refinement. And that contrast is really important here because it shows that the potential is there. This is a family full of dynamic plants that have been successfully developed into ornamental and economic crops. But Pycnanthemum or mountain mint is still at an earlier stage of that process. It's undomesticated. Now, even with all that diversity, there are some really consistent traits that define this family, Lamiaceae. One thing that you often hear associated with mints is that they're deer resistant. And sometimes that's true, but not always. And that's actually a good reminder that even with a family with shared traits, there's still a lot of variability in how those traits are expressed. One of the classic ones, of course, is square stems. If you've ever rolled a mint stem between your fingers, you can actually feel those edges. And another is opposite leaf arrangement, where leaves come off the stem in pairs directly across from each other. And these are really helpful characteristics for identifying members of the mint family in the field. And that brings us back to mountain mints. Because while all these share defining features, they also show a lot of variation. Beyond that, variation that doesn't always fit neatly into categories. How species relate to each other within Pycnanthemum is a much more complex problem. Now, we've talked a little bit about the family and the broader context. Now I want to actually introduce you to the plants themselves. These are all mountain mints. At first glance, they might look pretty similar. They all have these dense cluster flower heads, often with white or light purple flowers, that kind of silvery or minty green foliage. But when you start to look more closely, you begin to notice differences. Differences in flower color, spotting, differences in the size and shape of the bracts, differences in how tightly those flower heads are packed together. And these differences are what we often use to distinguish species. But the challenge is that these traits don't always remain very consistent. You can find populations that look like one species in one location, and then you can find something slightly different somewhere else, or plants that just don't seem intermediate between the two described species. So even at this level, we're not just looking at flowers. You can start to see that variability showing up again. And this is really about the question concerning species boundaries and where that question starts and becomes more complicated over time. So stepping back a little bit, I want to go ahead and give an overview of this genus. Mountain mints are native to North America, and they're pretty broadly distributed, especially across the eastern United States. They're also quite hardy across zones 4 through 8, which makes them really adaptable across a range of climates. In terms of diversity, there is roughly 19 to 25 recognized species, depending on how you define them and which taxonomist you're talking to that day. And that range is really important because it reflects some of the uncertainty within the genus. Different taxonomic treatments will split or lump species differently, which again points to those underlying complexity. Another key point is that these plants are cross-pollinated. They rely on insects for pollination, and they attract a wide range of pollinators. So when you combine broad geographic distribution, multiple species, and cross-pollination, you end up with a system where variation is not only present, it's expected. And that sets the stage for everything we see next. So this map by Bonet helps to visualize the distribution a little bit more closely. You can see that the mountain mints are connected primarily in the eastern United States, with a really strong presence across the Midwest, Southeast, and even up into the Northeast. However, there are a few species that don't always fit that trend. You may be asking yourself what's going on over there in California. That would be Pycnanthemum californicum. Even though there are species that go outside of those boundaries, like the californicum, the core diversity is definitely within the eastern half of the country. And this kind of distribution is important for a few reasons. First, it means these plants are experiencing a wide range of environmental conditions, different soils, climates, and elevations, and all those factors can influence how traits are expressed. Second, it means that populations are often geographically separated. So you can have local adaptation, where plants in one region look and behave differently from those in another. And when you combine those with cross-pollination, you get this really dynamic system where variation is constantly being generated and reshaped. So instead of thinking about mountain mints as a set of fixed, clearly defined species, it may be more accurate to think of them as a continuum of variation across space. And that's exactly the challenge and opportunity. And this project is trying to address. So, one of the reasons that this plant is so interesting is its whole range of habitat. On the left, here you have a granite outcrop where these plants can inhabit. These environments are really extreme with shallow soils, high heat, and limited water availability. Next, you can find them in remnant prairies, more open systems, and these are more open systems, often with deeper soils and a different set of competing species. And then you can even find these in disturbed areas, roadsides, field edges, places that are constantly being altered. Mountain mints show up in all of these. So right away, you're dealing with a plant that is incredibly adaptable. But that adaptability also comes with variation, of course. Plant growing on a granite outcrop may look very different from those in a prairie or along a roadside. Differences in size, leaf thickness, growth habit, and even flower timing can be affected by those environmental factors. And that raises an important question. Are these differences environmental responses, or are they genetically distinct populations when we're looking at variation across this genus as a whole? And that's a really hard thing to tease apart. But it's also a key piece of understanding how diversity is structured within this genus. So, next time you're out on a hike, or honestly even just walking along a roadside, I want you to keep an eye out for plants like this. Because once you start noticing mountain mints, you realize that they're actually pretty common, especially if you're anywhere in the southern Appalachians. They're not rare plants hiding in isolated locations. They're part of the everyday landscape. But at the same time, most people don't recognize them. Even fewer people think about them as unique individuals across their range. And that's part of what makes this group so interesting to study. They're accessible. You can see them in the wild. You can collect them from different populations. And you can observe how that changes across environments. And yet, there's still a lot we don't fully understand about how those populations relate to each other. So this isn't just a greenhouse or lab-based system. This is something you go out and see in real time. So despite all that complexity, mountain mints are already starting to become used in a lot of different ways. You can see them incorporated into flower arrangements. They have this really nice texture and structure that works well in mixed bouquets. Here you can see a more educational or interpretive setting. Botanical gardens will often display them in places that are more accessible to folks to help teach them about native plants and pollinators. And you can also see them used more broadly in landscape design and prairie type plantings. Of course, one of their biggest uses is ecological. They're incredibly valuable for pollinators. But what's interesting is that even with all of these uses, they're still not widely developed as an ornamental crop. You don't see a huge number of named cultivars, and you don't even see consistent material in the nursery trade. And there's also still a lot of variability about how well they perform. So again, there's that whole gap we're trying to address. We know they're useful, we know they're valuable, but we haven't fully bridged the gap between wild diversity and reliability. So one of the reasons mountain mints have gotten so much attention is because of how well they support pollinators. Here we have just a quick snapshot of what that looks like in real time. Multiple insects visiting the same plant all at once. This is really typical behavior for mountain mints. When they're in bloom, they're constantly active. If you walk by this plant in the field, it is practically vibrating. But even what's more striking about that actual quantity is that there's data to back all of that up. So this here is a 3-year study looking at pollinator visitation across different plant species. And what you see is cluster mountain mint is not just doing well, it's outperforming the competition. Over 4,000 visits were recorded compared to the next highest of around 900. So this isn't just anecdotal. This is a consistently high-performing plant for pollinators. And that's a big part of why there's so much interest in this genus. Because if we're thinking about designing landscapes that support biodiversity, uh, plants like this are incredibly valuable. But again, there's that whole tension. We have something that's clearly important ecologically, but we still don't fully understand that diversity within it. And that's where my work starts to become really important because we're thinking about conservation and use. And that use has really started to gain a lot of interest from those in a more broad context. Pycnanthemum unicum, or clustered mountain mint, was named the 2025 Perennial Plant of the Year. And that's a big deal in the horticulture world. It means that this plant is being recognized for its performance, its adaptability, and its value for the landscape. So on one hand, we're seeing an increased attention and promotion of this genus. It's becoming recommended for gardeners, for pollinator plantings, for sustainable landscapes. But on the other hand, most of that attention is focused on just a few species like Muticum, and the rest of the genus is still relatively unexplored. So while one species is gaining recognition, there's this whole broader group of plants that we don't fully understand yet. And that raises an interesting question. Are we only seeing the tip of the iceberg in terms of what this genus can offer? Now, this is where everything really starts to come together. So when you start looking at this plant across different populations, different species, different environments, you see just how much variation there is. You'll have variegated varieties like this, and you'll even have yellow-colored varieties like that. This variation isn't just between species, it can occur across within species, within populations, or even what we think is a single taxonomic unit. So now we're back to the core question. What actually defines a species in the genus? Is it morphology? Is it geography? Is it genetics? Are we looking at something more continuous, something that doesn't fit neatly into discrete categories? And that's really the challenge of this work. Because this variation is both the problem and the opportunity. Although it makes classification difficult, it also provides raw material for selection, improvement, and discovery. And that's where the next part of this project really begins. Now, I want to go ahead and push the idea of variation with just another picture. Because it's not just about different populations that look different. Sometimes you see really unusual forms, even what we think is a single species. Here we have Pycnanthemum tenifolium that looks almost exactly like a creeping juniper. How insane is that? It's even more compact, almost mound forming, with a really fine texture that's very different from other members of the same species. And when you see something like this, it raises a lot of questions. Is this just environmental? Is it genetic? Is it something stable that could be selected and propagated? Or is it just one-off expression? And this is where things start to get really interesting from a breeding perspective. Because variation like this might represent exactly the kind of material we're looking for. Traits that could be useful in ornamental development, but without understanding the underlying biology, it's hard to know exactly what's happening here. So again, variation is everywhere, but interpreting that variation is the main challenge. So coming back to our story again, now with a little bit more context, we started off with that simple question, what is a mountain mint? And now we've seen that even at that level of growing plants and observing traits, that question starts to become even more complicated pretty quickly, because that variation doesn't always match our expectations. And that leads us to, oh, really, what is happening with this genus? Why do we see so much inconsistency? Why are traits not lining up cleanly as with the species descriptions? At this point, we looked at morphology, habitat, distribution, but those only get us so far. So let's start to really answer the question. So one key piece of this puzzle is looking at ploidy and genome size. Now, ploidy is the number of chromosomes within a specific plant. Now, a lot of the foundational work on Pycnanthemum cytogenetics, or what makes up this plant genetically, is actually quite old. Some of this work is going back 50 years. And what we know is relatively low. Our labs have been able to work out the genome size of the genus of about 2.89 to 7.2 picograms, which is a pretty big range. And this suggests a pretty big difference in ploidy, differences in the number of chromosome sets within these plants. But the problem is that the data is incomplete and sometimes inconsistent. A lot of these plants are polyploids, meaning that members of the same species can have multiple sets of chromosomes, which makes breeding a little complicated. And it's not always clear on how we understand how those species relate across species because of that. So we're left in a situation where we know ploidy is important, we know it likely plays a role in variation, hybridization, even species boundaries, but we don't quite have a clear modern framework for how all of that fits together. There's a major gap. Because if you want to understand how these plants are related, how they evolve, how they might use them in breeding, we need to understand a pretty clear idea of their genomic structure. Now, before we go any further, I want to quickly define a few of the traits that are commonly used in distinguishing species within this group. I'm going to pay particular attention to the Virginiatum clade or group of plants within this genus. So on the left, we have leaf size, which is typically measured from tip to base. This is one of the most straightforward traits that we find within the plant. Now we have trichomes or plant hairs, and that is a common trait within the mint family, especially. And it's an easy way to identify the plant when there isn't traits present, but specifically where they are. And when talking about where that trait is present within the plant, we are talking about the face or the flat surface, and then the angle or the edges of the plant. And then we have calyx length, basically the base of the flower, which can vary depending on which taxonomic key you're using. So in theory, you can measure these carefully and you should be able to distinguish species. The key phrase here is in theory, because once you actually start working with the plants, things don't always line up that clearly. And that is often why this plant is given a range rather than an exact number of species within the genus. So here I'm going to go ahead and show you 3 different species: Pycnanthemum virginianum, verticillatum, and Torrey. Now we're going to get to Torrey a little bit more specifically in a bit, but here I want to go ahead and start to show some of the morphological overlap within the genus. Within the genus. So here I have a few traits listed: leaf size, trichomes, and then also calyx. And now when you look at those numbers, there is a lot of overlap. Leaf size overlaps, calyx length overlaps, even trichome placement can vary. So now instead of having clear discrete categories, you start to have ranges that blur into each other. And we're talking about the difference of millimeters here. That makes identification really difficult, because if you have a plant that falls right into the middle of those ranges, it's not always clear which species it belongs to. And that is exactly the issue people run into when working with this genus. The traits we rely on are variable. They don't always separate species cleanly, and that raises a bigger question. Is this just natural variation within a species, or something else happening here? And more importantly, this can complicate conservation decisions when we have one species that has high conservation value. And something else may be happening here, right? Hybridization may be happening. Now, this figure comes from earlier work looking at artificial crosses between the different Pycnanthemum species to see just how well they hybridize. And what it shows is that many of these species are actually capable of crossing with each other. That means they're very promiscuous. These are connections between multiple species, suggesting that reproductive barriers may not be as strong as we might usually expect. And here we have an example of how we're starting to explore that experimentally. This is a pollinator cage setup. Basically, this is allowing controlled crosses between plants while we're still using natural pollinators. So instead of hand pollinating everything, we can let insects move pollen within a defined space and see what combinations actually occur. And that's really important here. Because if species are infertile, if they can hybridize and produce viable offspring, then that could explain a lot of that morphological variation we're seeing. And it would mean that instead of distinct isolated species, we may be dealing with a network of interacting populations. And that has some big implications, especially for breeding. Because hybridization uh, can vary and complicate identification, this is really where the story starts to shift from describing variation to understanding the processes behind it. So one way that folks have been able to deal with this kind of complexity in other mint groups is through core collections. Now, for those of you who are not familiar, I'm going to go ahead and talk about the Mentha core collection. So this is another genus in the same mint family, probably one that you're all familiar with. And this takes a pretty big idea from a global collection of accessions and distills it down into a representative subset, usually around 10%. Now that core collection, or 10% of that genus, captures most of the genetic diversity. More importantly, these collections often include hybrid taxa, multiple species, and material from across the world. So instead of trying to force clean boundaries between species, they embrace the complexity. And this allows researchers to ask bigger questions about diversity, relationships, and trait variation. It also raises a pretty big question about Pycnanthemum. What would it look like if we took a similar approach to a USDA core collection? Now, before I go any further, I want to go ahead and highlight one really neat thing about these USDA collections, germplasm collections specifically, is that all of this is material that is free to use for researchers. We can request seed from all around the world and get it delivered right here to our lab. And that is essential to what we're starting to do. Now, this is our common garden setup at the Durham Horticulture Farm. I showed y'all it earlier. And each of these plots represents a different accession collected from wild populations, herbaria, and cultivated sources. And by growing everything in the same environment, we can control how much environmental variation there is. We're essentially creating a closed system here. And when we see differences between those plants, we can have a lot more confidence in how those differences are genetic or environmental. And this is really important for a genus like this, because it allows us to directly compare morphology, growth habit, and performance across a wide range of material. So within that common garden experiment, we're measuring a range of traits. On the qualitative side, things like flower color, maculation, or pigmented spotting on the floral lobe, pubescence, and of course, pubescence. These traits are often used in taxonomy and very helpful when identifying a species. Then on the quantitative side, we're looking at things like plant height, crown area, growth index, SPAD readings, which is essentially how much chlorophyll the plant is taking in, and also flower production. We're also tracking things like defoliation and lodging using standardized rating scales. And that gives us a lot more of a complete picture of what's actually happening with each plant. Not just with how it looks, but how it performs, and that's key. Because if morphology alone is enough to separate species, maybe combining morphology with performance data can help us better understand patterns across the genus. And what did we find here? And this is a bit that's very helpful for gardeners when choosing what plants to have in their garden. So starting off with the qualitative traits, what you see is a whole range of traits across different species. And a few things start to become really clear. First, most species are white flowered. There are a few exceptions with purple or purple to white variation, but overall, flower color is not doing a great job separating them. Second, floral spotting is almost always present, and that's that maculation. So again, not very informative for distinguishing taxa. And then pubescence. And this is where things start to get a little more interesting. We do see differences in where trichomes occur on the faces versus the angles, but even here, there's still a lot of overlap across the species. So the takeaway is, even though when we scale up and look across the whole genus, the same problem holds. These qualitative traits don't clearly separate the species. Now, something that I wanted to draw back earlier is I showed you a yellow flowered species. That is relatively new to us and something we haven't been able to include in the garden evaluation study. So, regarding the quantitative traits, when we look at these quantitative traits, we're looking at the patterns that emerge, and then we're looking at performance. And the answer is yes, but not in the way that you might expect. Instead of traits grouping by species, we see different species excelling in different traits. For example, Pilosome shows up repeatedly in crown area, flowers through stem, and growth index. Verticillum and Torrey also perform really well across multiple categories, but no single species dominates everything. Instead of clear species boundaries, what we're seeing is functional diversity spread across the genus. Different species are good at different things here. And this is where everything starts to become really useful. Because when we're using this information from the garden evaluation study or the common garden study, it is helping us to inform our breeding perspective. We don't necessarily need perfectly defined species, but we need our plants with desirable traits. So now instead of asking, what species is this? We can start asking, what does this plant do well? What species have high flower production? Which ones have better structure, less lodging, or better foliage retention? And that shifts the framework entirely. We move from trying to force clean taxonomic boundaries to using variation across the genus as a resource. And that's where the system starts to become really powerful. And that leads us back to where we started. At first, it was a simple question, what is a mountain mint? But pretty quickly, that became a more complicated question when we started to look at variation. And now we're going to start to look at it as a conservation problem and the breeding potential with that. So one species that starts to sit at the center of this is Pycnanthemum torreyi. Now, torreyi is a cross-pollinated species. Like many in the genus, it shows variation in ploidy. Both tetraploid and hexaploid populations have been reported. That is 4 sets and 6 sets of chromosomes, respectively. So even within a single species, there's still already genetic complexity, and that makes it a really interesting system, both for understanding relationships within the genus and for thinking about how we might use the variation moving forward. Now I want to take a moment to break down the name of this plant. Pycnanthemum roughly translates to densely flowered, which you can see here in those tight clusters of flowers. Now, as many of you may be familiar, Torrey is actually named after John Torrey, one of those early American botanists. So there's this direct connection between historical collections and modern research, and that's important. A lot of the material we're working with today traces back to those early efforts to document plant diversity. So in a way, this project is building on that foundation, but using new tools to ask the same fundamental questions. What exactly are these plants, and how are they related to each other? Now I want to take a moment to acknowledge John Torrey's contribution to my work. As many of you know, he was the founding botanist in North America. He described and named a huge number of plant species, and his work really laid the groundwork for how we understand North American flora today. On the right is one of his herbarium specimens associated with his work, and this is really important because historically, species concepts were built from a limited number of specimens like this. So when Torrey described Pycnanthemum torreyi, he was working from a snapshot, a single representation of what he thought that species looked like. But now we have access to many more populations across a much broader geographic range. And that's where things start to get complicated. Because when we look at beyond the original specimen, those boundaries of this species start to become much less clear. Now, despite that long history of study within this species, starting with John Torrey, much of this species' native range started to face significant declines, in some areas of up to 50 to 70%. So what we're looking at here is not just variation across whole, it's decline that isn't quite uniform. Uh, as you start to see that decline narrow down even further, you start to see what's left. In some regions, we're down to just a handful of remaining populations, sometimes as few as 4 to 12 different populations. So what was originally a pretty broadly distributed species from northern Georgia all the way up to New Hampshire, west out to Kansas, has now dwindled to about to a few remaining populations in Virginia, Kentucky, and possibly North Carolina. And in particular, these are wild populations, not those found in cultivated or garden settings. So at this point, we're dealing with a pretty clear problem here. We have limited resources, declining populations, and not all populations are equal in terms of genetic diversity. So conservation starts to become a question of prioritization. Which populations are we going to protect here? And which ones hold the most variation? And which ones are at the risk of being lost? And that's where our barrier starts to become really powerful. That gives us access to specimens we've collected across space and time, often going back more than a century. We relied on herbaria like UNC Chapel Hills, New York Botanic Garden, and my very own Georgia Herbarium. What we're doing here is we're physically sampling leaf tissue from herbaria specimens, typically small amounts, around 50 milligrams, and then about 100 milligrams for fresh tissue. Now, this has to be done really carefully. These specimens are often irreplaceable. So what we want to do here is minimize the damage while still collecting enough material for DNA extraction. And this is a really hands-on process. We're going through specimens, selecting appropriate tissue, and preparing samples for extraction. And this is where the historical archive starts to become something very tangible. You're literally holding a piece of a plant collected decades ago and preparing it for genomic analysis. So here we start to begin to address the gap. I set out to assess the genetic diversity and population structure within Torrey. And to do that, I assembled a data set of about 90 different specimens spread across 15 states. And this includes a mix of herbaria material, wild collected samples, and nursery accessions. And what we're capturing here is both historical and contemporary variation. That is important. The data set includes specimens dating back as far as 1863, allowing us to incorporate long-term perspective on diversity of this species. Together, this provides a broad foundation for understanding how variation is distributed across the whole species. Now, when it comes to using herbarium DNA, there are some challenges. When we are working with herbarium specimens, we're not just dealing with fresh, high-quality DNA. We're working with material that is often decades, sometimes even more than a century old. Over time, DNA degrades. It breaks into smaller fragments and becomes chemically modified. At that point, it can even become contaminated by fungi, bacteria, or just even environmental exposure. In particular, when you're dealing with some of these older specimens, they might have been treated with different chemicals that have just totally destroyed the DNA. Those chemicals include flora, and those chemicals used vary quite a bit. So instead of a nice, clean, high molecular weight DNA that we can usually get from fresh specimens, we're often working with something that is fragmented and damaged, and that has real consequences. It affects how much DNA we can recover to use in our genomic analysis, and that also affects sequencing success. You really want to capture that diversity here within this species, because it helps us inform conservation efforts and then also potential breeding efforts. Now, understanding that, if we do have DNA that is poor, that can introduce bias downstream during analysis. So while herbarium genomics opens incredible doors, it also forces us to be very intentional with how we extract, process, and interpret that data. And again, this is material that more often than not, you're only going to be able to sample from once. So when you do perform that extraction, you want to make sure you are being as careful as possible. So to work around these challenges, we relied on protocols that were also specifically designed for degraded plant material. For this project, I used a modified extraction protocol with a plant and fungal DNA kit, and that is pretty well suited for dealing with secondary compounds and low-quality tissues. And the key here is to be gentle but efficient. You want to maximize DNA recovery without further damaging what is already fragile. From each specimen, we take a very small amount of tissue, like I said before. And with that small amount, we're able to recover enough DNA to move forward with sequencing. So what is really exciting here is that even with these historical specimens, things collected over 150 years ago, we can still contribute to modern genomic data sets and help inform our decisions today. And that starts to become they start to become those active data points in understanding genetic variation. Now, unfortunately, I don't quite have that sequence data back yet. But once I have that data and that information, the next step starts to become, what are we actually trying to learn here? And at the most basic level, we want to know whether populations of Pycnanthemum torreyi are genetically distinct from each other. Are we looking at clearly separated groups? Are we looking at continuous variation across the landscape? We're also interested in how these plants, whether or not these plants are so-called hybrids, how there are individuals that do not fit cleanly into the existing species descriptions, and for those individuals that don't fit clean into those descriptions, are they true hybrids between other members of the Virginianum clade? Or are they actually stable, consistent lineages that just have not yet fully been recognized? So really, is Torrey a mix of different species? We don't know. And finally, and maybe even more importantly, from a conservation standpoint, we want to know what we should be prioritizing. And some populations containing unique genetic variation, those may be disproportionately important to conserve. So this is where the data starts to become really actionable. It's not just about describing variation, it's about using that variation to guide decisions in both conservation and breeding. And that leads us to the whole breeding question. Now, should we mix populations? And there's not really a simple question here. There's not really a simple answer here. On one hand, yes. Mixing populations can help rescue declining species. If populations are small and isolated, introducing new genetic material can increase diversity and reduce inbreeding. It may also help preserve variation that we do not fully understand yet, what is often referred to as hidden diversity. And from a restoration standpoint, it could give us a more resilient plant material to work with. But there's also a strong argument for no. Mixing populations can disrupt natural selection processes. These processes have been shaped for hundreds of years and have affected these populations for really long periods of time. And it can essentially create what I like to call curated genotypes. These are things that don't naturally occur in those environments, and most importantly, it can break local adaptation. Populations may be uniquely suited to their specific conditions, and mixing them can actually reduce fitness rather than improve it. So this is just not a genetic question, it's actually an ecological and an ethical one. And it is exactly why understanding population structure using genomic analysis matters so much. So where does all of this work lead? What I find really exciting about this work is that it just does not sit in one field. It connects to a wide range of people and applications. For conservationists, this work can help identify which populations are most critical to protect. For restoration practitioners, it can guide sourcing decisions, what material to plant and where. For breeders, it opens the door to selecting traits while still maintaining genetic integrity. Even for land managers and policymakers, it provides a more informed foundation for decision making. So what starts as a question about a single species actually starts to become something much broader, a framework for thinking about diversity, adaptation, and how we manage plant populations moving forward. That is really where I start to see this work going. Now, I want to go ahead and take a moment to acknowledge all of the folks that have helped me with my work. I want to thank my advisor, Dr. John Reuter, for his guidance and support throughout this project. And I also want to thank my lab mates and research tech, Greta Galina, Trent McCracken, respectively, and Juliet Ane. And a huge thank you to all of the contributing Iberia for my work and that contributed material. This project would not exist without them. And then, of course, the different funding sources that have helped with my work. And with that, I want to say thank you and open the floor to any questions.
Thank you so much. That was great. Um, for our folks in the audience, if you want to unmute yourself, feel free to do that. If you'd like to put your question in the chat, feel free to do that. While people gather their thoughts, I have a question. What do you have a favorite species of mountain mint or species? Species.
Yes. So the question was, do I have a favorite species? And of course, I have a favorite species. One one in particular that I find incredibly charismatic is Pycnanthemum montanum. Unfortunately, I'm not quite sure of what that common name is, but you will see such a large amount of variation within that one species. You see white flowers, yellow flowers, a high amount of anthocyanins. Um, that makes red in the foliage. It's also one of the few species within the genus that you can actually find on your way when you're just walking along the trail on a hike. You'll see it in an understory in complete shade, and that's what makes it a little unique compared to the others.
Thank you. Nan, I see you have your hand up. Would you like to ask a question?
Yes, yes, thank you. That was absolutely fascinating. Yeah, I hope you can hear me. Yeah, I work in a native plant nursery here in Virginia. I'm also really interested in the wild populations and all of the research that you talked about. Um, it's interesting because, of course, there's a push there's a push to have, straight species in the nursery. I mean, it depends. And I often think it depends which kind of client you're dealing with in a suburban landscape, like, I don't know, does it matter? But there are many, many people who are really looking for straight species. And it's funny because I think of mountain mint, as you say, we don't have cultivars of it. We just have several species of it. And it's such a delightful plant too. When people are at that place where they're ready for a little wildness in their gardens or they're, have that kind of landscape where they can bring in that wildness and support like all those pollinators and all those bees that they're going to. It's really exciting to introduce them to mountain mint. And I guess I was wondering, it's such a great straight species, but do you really, I would love to learn more about how you feel like we need to maybe have more domestication in mountain mint specifically for the trade.
Yeah, absolutely. So the question was, I suppose, my thoughts and feelings about plant domestication of this genus as a whole and how that can be applied. Um, and it is definitely a difficult question, especially when there's conflicting views about how we should view native plants. Um, my prerogative, my goal is to make these plants more accessible. And that accessibility means making cultivars that are fitted towards a wide variety of landscapes that can also fit well in those wide variety of landscapes. I want this plant that is so good for pollinators to be in everyone's garden. And to make that possible, that means curating a genotype, so to speak, and improving the habit, making sure the flowering is more uniform, that sort of thing. Now, that doesn't have to be applied to every member of this genus. We can leave the really rare species alone. We don't have to work with them. There's still
Mhm.
plenty of variation across this genus as a whole.
Can I follow up then? So would you suggest that with Torrey, you would not maybe would you have more qualms about domesticating that or working with that till you because it is so rare already? I'm just curious.
It so the question was concerning breeding Torrey specifically, and it's rather complicated. We're not quite sure yet what the diversity within Torrey looks like. So breeding with it from a more practical standpoint may just be that difficult. Um, and then it the question here, I think a better way to look at it is what exactly would we be breeding it for? Would we be breeding it in sort of a conservation perspective? Are we looking at heterosis or hybrid vigor with crossing it with another species to help improve the plant and make it more competitive against invasives? Or are we breeding it for a landscape? And that's where it starts to get a little tricky. Um, I don't
Thank you. That's fascinating. Yeah.
Yeah, of course.
All right, we have a question. We have a couple of questions in the chat, but first from Casey is, what species concept do you find best fits with mountain mint?
So the question was, what species concept do I think best fits with mountain mint? I'm not entirely sure what you mean by species concept.
Oh, sorry. I was in public before I had to type it out. Yeah, so there's like biological species concept. Like you could define species just based on genomics, et cetera, et cetera. Like
Mhm.
ecological species concept.
Um, the short answer, well, it's the short answer to a long one is um there is so much morphological variation across the species. It's really hard to tell just from a key what different species are. Um, the next best step is going to start to use your genomic tools. So, for example, I'm using GBS for Torrey, or genotyping by sequence,
Okay,
and that is a pretty easy way to understand diversity.
cool. Nice. I'll look forward to those results.
Yeah,
Thank you.
absolutely.
Um, okay, also in the chat, Joanne asks, are Monarda species as confounding as mountain mint?
So the question was, are Monarda species as confounding as mountain mint? Um, Luckily, I only work with one member of the mint family, so I'm not really sure. If Monarda is like any other member within that family, the answer is probably yes. But
Yeah.
I'm not sure.
Maybe someone else in the chat has some Monarda insights. Um, from Allison, if you hybridize species, does that not mean that seeds from those hybrids would be sterile?
Um, so the question was, if we hybridize these species, does that mean the seeds from those F1s are sterile? Um, s- Not always. Um, when we hybridize these different species, they can be with different ploidy levels. So more often than not, if you take a plant that is diploid or has two sets of chromosomes with a plant that has four sets, and when you cross those two species, you can make a triploid, meaning that it has three sets of chromosomes and it's uneven. So more likely than not, that plant is sterile. But if you cross a diploid and a diploid, you can produce other diploid offspring, meaning they have an even set of chromosomes, and those plants are still fertile. And across the species, there's a pretty wide range of ploidy levels. So multiple species are diploid, and unfortunately, a lot of those species are polyploids, meaning uh, not just one ploidy level is assigned to one species. There can be multiple ploidy levels. So the answer is yes and no. That's the short answer.
Okay, I want to give folks just one more moment for any last minute questions. We're a little after 7, and then I have a final question that I like to end with. Um, okay. I don't hear anyone unmuting or typing away in the chat. So, uh, Tanner, if there's one thing that you like would want the audience to take away from your talk, if there's one thing that you want us to remember and like take on into our lives about mountain mint or about how we think about species or cultivating these plants, um, what would that thing be? What's
One takeaway that I would really like to leave y'all with is the importance of pollinators in our habitat, and more
Mm,
importantly, how native plants interact with those pollinators. We have such an amazing range of diversity within our own backyard of different species that are native to the US that we can play with, hybridize with, and that are so great and fit so well in our landscapes. We just have to change our approach to how we use them.
Thank you so much. Thank you everyone who joined us tonight. For more information on the Torrey Botanical Society, please visit torreybotanical.org and consider becoming a member. And have a good evening. Thank you, everyone.
Thank you.